I believe that people here, we feel very honored to work here for a company like this, like Fresenius Kabi. The passion that you can feel it, you can actually touch it when you're walking through the hallways and everywhere in the facility.
1, 2, 3.
We like to invest in our people because we want to assure that we are manufacturing products with quality.
People have to think in quality, and they will do quality.
People really know, because we insist on teaching the people the use of the product, that we save lives.
Y para mí ha sido una experiencia maravillosa, ya que trabajamos con productos para salvar vidas. Carry for life.
Greetings and welcome to our overview of plasma. I'm Amy Fontes, President and CEO of the Plasma Protein Therapeutics Association. This is intended to serve as an introduction to how plasma is made into life-saving therapies, to address the conditions they treat, and to discuss plasma's role in treating COVID-19. We're fortunate to have with us today an impressive and esteemed group of individuals, each of whom has a distinguished career in the field of plasma. They include Bill Zabel, President of North American Sales and Commercial Operations for Grifols and Chair of the North American Board for PPTA, Dr. Milan Novakovic, who heads medical for Takeda in Germany, and Dr. Deborah Benson Kennedy, Medical Director for CSL Behring. Each of our panelists will offer a brief presentation to illustrate to us the different facets of plasma-derived therapies. And then when we're finished, we'll spend some time with Q&A to further interrogate the mysteries of these life-saving products. So let's begin with Bill Zabel, who will address the production process of PPTs. Bill?
Amy, thank you. So I've been asked to describe what is plasma, how is plasma collected and manufactured, and then I'll turn it to Debbie. Okay, so what is plasma? And you can see by this illustration here that plasma is the clear, straw-colored liquid portion of the blood that remains after red blood cells, white blood cells, platelets, and other cellular components are removed. And you can see in the description to your right there that plasma is the single largest component of human blood. It's comprised of about 55%. It's that yellowish portion you see on the right to the vial there, and contains things like water, salts, enzymes, and antibodies, and certainly other proteins. So how is plasma collected, and how is plasma manufactured? And you can see here that plasma can be collected in two different ways. I'll start first by mentioning something not on this slide. So plasma can be collected from whole blood. Some of you may recall some of the donor drives. I know in the United States here, it's the American Red Cross. And when we collect plasma from whole blood, that's called recovered plasma. In this illustration, and probably most common in your left-hand side there, is plasma is obtained through a process called plasmapheresis. And again, plasma is collected from healthy donors. And you can see in that illustration there that we take whole blood utilizing plasmapheresis. We separate the blood and the plasma, and then we collect that plasma that you see in that vial there, and then we return the red cells, the white cells, and other components to the donor. And if you take your eyes and go to the right there, that's just another illustration there that we now take plasma into the process here, where within plasma, you can see that only a small portion of that, and Debbie will mention this later, about 7% of plasma is those proteins that we actually take into our manufacturing process. This is an abbreviated illustration, certainly of this manufacturing process that takes 7 to 12 months. but we take and fractionate the product or the plasma, and that means we remove the different proteins or components. We purify that all this process here from healthy donation, certainly to the final container where it's sterile filled, we remove viruses, certainly enveloped and non-enveloped viruses from the process to get a series of proteins that you see illustrated in that pie chart. The most abundant protein we collect from plasma certainly being albumin. And we like this illustration on the bottom because this is a human donor. This is the vein-to-vein concept. So 7 to 12 months from the time we collect it from a human donor to the time it's actually infused within that patient we see on this slide that relies on these medicines. Now, Debbie's going to get into this with more detail here, but I wanted to illustrate that people living with these rare diseases use plasma proteins for a whole bunch of different things. I think most notably here that we want to point to things like you may be familiar with hemophilia. And if you look at the right, then there's an abundant need and a big call to action, certainly as we're within the latter parts of International Plasma Awareness Week, that we need people to donate plasma. So in this illustration to your right, you can see it takes 1,200 donors. That's 1,200 donors to treat just one plasma patient that relies on a treatment for hemophilia, and that's plasma-derived factor VIII. And I think for all your panelists and people associated with this industry, the reason we're here is because of individuals like this. So meet Terry here, an individual with primary immunodeficiency. And again, through our awareness, education, and commitment to making these plasma protein therapies, we want to make sure that patients like Terry have these medicines, in this case, intravenous immune globulin or subcutaneous immune globulin, when and where he needs it. And I'll close just by putting another face onto it. We know a lot of the parents of children with hemophilia. And this is just her credit to this industry and their commitment to patients that suffer with rare and serious disease. So I'm going to pause there, and I've had the opportunity to meet Debbie. She's got some exciting next steps in our story of plasma and the journey of plasma here. So Debbie, I'm going to pause here and turn it over to you. And thank you for this opportunity.
Bill, thanks for that thoughtful presentation. I know I have questions for you about the manufacturing timeline, and I look forward to our Q&A. Next, we'll hear from Dr. Deborah Benson-Kennedy from CSL, who will talk about the increasing clinical need for plasma protein therapies.
Thank you, Amy, for that kind introduction. So I am fortunate today to speak to you about what is in plasma. Plasma is composed mostly of water, and there are many different types of proteins in our plasma, and this is what we're going to speak about for the rest of this talk. But as Bill shared with you, it's only 7% of our plasma. And about 1% are our minerals, salts, sugars, fats, hormones, and our vitamins that are carried within our circulatory system. But before I start speaking about what's in plasma, I want to remind you that donated plasma cannot be made in a laboratory. The pharmaceutical industry is able to make many drugs from a chemical standpoint and be able to manufacture that in a plant without any need of human components. However, plasma proteins, the best manufacturer is the human body. So what types of plasma proteins are in plasma and what conditions do they treat? So I'm going to go through a number of these conditions over the next coming minutes. I'm going to begin with immunoglobulins. Well, what is an immunoglobulin? These are the proteins that we also call antibodies. And there are many disorders in which individuals cannot make appropriate amount of antibodies, and some don't make antibodies at all, which makes them immunodeficient and subject to very high risk conditions and diseases. It is absolutely necessary for these individuals to receive antibody replacement therapy to keep them safe and free of infection. Antibodies can also be used as immunomodulators. So people that have overactive immune systems, there are times in which immunoglobulins can help slow down that process and treat disorders of chronic inflammation. Many of those are in the neurological disorders, and I'll speak to those also in a few slides. But the pie graph to the left shows that there's many medical uses for immunoglobulins and how critical these are for very rare and serious disorders. Only about half of our immunoglobulin and antibody therapies used are for the conditions of antibody replacement and neurological disorders. Speaking a little bit more about primary and secondary immunodeficiencies, these diseases are extremely serious, and people are absolutely dependent on receiving antibody therapy to keep them free of infection. And as Bill alluded to earlier, I want to once again remind you how many donations are necessary in collecting plasma to have therapy available for a single person for 1 year. It's approximately 130. I cannot emphasize enough how important continuous donation of plasma is needed to make sure that we can continue to manufacture adequate amounts of product for individuals that need them. The neurological conditions that are commonly treated with immunoglobulin are also extremely serious and quite rare. However, diseases like multifocal motor neuropathy, Guillain-Barré syndrome, and chronic inflammatory demyelinating polyneuropathy can be incredibly debilitating, and the need for immunoglobulin is quite high. These disorders can impact individuals' motor function, keep them losing their ability to walk or move, and in the setting of Guillain-Barré syndrome, can cause complete paralysis, so they're unable to breathe independently. And this is a critical illness and can lead to death without appropriate treatment. But there are also specialty kind of antibodies that can be manufactured, and these are called hyperimmunes. We'll speak a little bit more about these with respect to COVID-19. But what I can share with you that there are hyperimmunes that have been used for many years in medical conditions. And one of the more serious ones, which you may be familiar with, is rabies. There are no known treatments for rabies. However, if somebody has had a rabies exposure, they can receive a hyperimmune and hopefully potentially save their life. So moving past the immunoglobulins, there are many other proteins that are life-saving for individuals with rare and serious conditions. One of these is alpha-1 protease inhibitor. This is an enzyme that keeps the integrity of the liver and the lungs intact. Unfortunately, there is a disorder where people inherit a deficiency in alpha-1 antitrypsin and require lifelong replacement. This is a plasma protein, and this can be administered to patients to help decrease the progression of their disease. Another inherited disorder which can be treated with plasma protein therapies is hereditary angioedema. This is a serious and potentially life-threatening disorder where individuals spontaneously begin to swell in any part of their body. This can cause pain and suffering, but the swelling can also happen within the throat and the airways, such that someone can have asphyxia, and this can be life-threatening. C1 esterase inhibitor can be administered intravenously and subcutaneously to not only treat hereditary angioedema, but also can be used chronically to prevent attacks. And coagulation factor disorders. As Bill alluded to, hemophilia, for example, is one of the more known, though rare, bleeding disorders, but there are many deficiencies in coagulation factors that are inherited and cause very significant and potentially catastrophic and life-threatening bleeding. All of these factors noted here can be isolated from plasma and concentrated and used to not only treat bleeding, but to prevent bleeding long-term for individuals that are treated, that need treatment for this disease. And last but not least, the most abundant protein that's in the plasma, albumin. Though this is a relatively inert protein, it's incredibly important and useful in the treatment of critical care individuals in the hospital setting. For many years, albumin is used to support individuals in burn units, organ transplantation, severe liver conditions, shock, trauma, and so forth. And this is an essential protein that's needed to help the hospital workers and individuals under critical illness in the ICUs. Though I'd like to thank you for your time and your interest in joining us today, but I'd like to also remind you how critical it is that we continue to encourage plasma donation and that our manufacturers continue to produce these incredibly important plasma proteins. I look forward to the Q&A, and I hope that you've learned something with this session. Thank you.
Thank you, Debbie. Plasma protein therapies serve such an important function, and there's so much exciting potential. Your presentation certainly highlighted that. Finally, we'll hear from Dr. Milan Novakovic from Takeda, who will talk about how plasma is being used to treat COVID-19. And this is something we're all interested in. Milan?
Thank you, Amy, for that kind introduction. After Bill and Debbie elaborated on the principle of plasma, I will take the therapeutic perspective with a recent example with convalescent plasma against COVID-19. I'm Milan, and I'm a COVID-19 Plasma Alliance member. What is convalescent plasma and how can it be used therapeutically? Basically, each patient which recovers from an infectious disease can be a convalescent plasma donor. Those who survive infections have useful antibodies. So how can these antibodies be at the end therapeutically used, either directly for infusion or by a stepwise process enriched on a specific antibody? And as you have heard by Debbie, we call them hyperimmunes. So there is a lot of historic example on this one, specifically treating infectious disease where no standardized or established drug treatment existed was a quite useful way to use convalescent plasma. And I give you a short overview on the next slide, because it started back into 1890 with Emil Behring and his collaborator Shibazaburo Kitasao here in Berlin. Basically, their seminal paper on treatment of diphtheria with convalescent plasma won Emil Behring the Nobel Prize in 1901. And over the time, and you see this here, multiple diseases of infectious nature have been treated successfully, starting with the Spanish flu in the 1980s and 20s, up to scarlet fever and common diseases like measles, varicella, up to hemorrhagic fevers like Ebola and Yunin fever, another form of hemorrhagic fevers, successfully, but never in the way that we are going to describe. And specifically in the 2000s, the experience or knowledge for acute respiratory diseases on influenza virus, but as well on COVID was existing. What is the difference between convalescent plasma and hyperimmunoglobulins, basically? Let's look first on convalescent plasma. It can be directly transferred from patients to people, and specifically, it's given on a short-term usage. By hyperimmunoglobulins, the difference is before they can be given therapeutically, they need to be processed in order to receive the status of a hyperimmunoglobulin-based drug. So two different processes, two different timelines. So on convalescent plasma, you give it directly. In the hyperimmunoglobulins, the donated plasma needs to be pooled and processed and specifically intended on a longer term perspective. Next slide, please. So convalescent plasma has, as of today, limited virus inactivation. You need to make sure that plasma from different humans needs to be safe as a therapeutic option. Therefore, you need to inactivate those viruses on site in the clinic. Each clinic has their own protocols. versus hyperimmune globulins, there is a comprehensive virus inactivation process. Very diligently, as mentioned, it follows the standard of a drug. One of the key differences between convalescent plasma and hyperimmune globulins, there is a variability in the concentration of antibodies because convalescent plasma comes by donors and therefore the inter-individual variability versus a constant antibody potency based on the sampling process and the purification. The therapeutic consequence is that you have a less strong antibody concentration in convalescent plasma versus hyperimmune globulins. There is stronger antibody concentration. There is more antibody in a smaller volume to be infused versus convalescent plasma. Next, we as an industry, we sat together because we had to react on this global threat. And within the global COVID-19 Plasma Alliance, we decided to pool our resources for the benefit of people around the world with this devastating disease. That's the reason why we work closely together with our goals to accelerate the development of a potential treatment, to improve change of success, and to increase, of course, the supply of this potential treatment. Thank you for the opportunity to be part of today's event, and I give it back to Amy.
Thank you, Milan. It's fascinating to learn the distinction between uses for convalescent plasma, and I certainly have more questions for you. So now that we're all a little more familiar with plasma and the therapies that plasma provides, I'd like to ask a few questions of our panelists. So Bill, we'll start with you. You talked about the production of plasma therapies and said that it can take up to a year. Can you elaborate on why it takes so long compared to other medicines?
Yeah, no, Amy, great question, and thank you. And oftentimes we're asked that, what's the difference between a plasma-derived protein being developed and manufactured versus a pharmaceutical? I know from my days at Cipro, if we needed more Cipro for an anthrax scare, we could do that overnight. I think most notably what we've outlined here, which is something we want the audience really to reflect on, is the starting material for a plasma-derived protein therapy is a human donor, a healthy human donor. And from that, of course, with a healthy human donor, we have to make sure that that plasma is appropriately tested. It's held for a period of time before it ever introduces the manufacturing process. We like to say the process is the product, and over the course of time, months, it goes through this long process of fractionation, which I mentioned before, purification, again, being removed with viruses along the way, both enveloped and non-enveloped viruses, before finally it reaches a sterile filling session where it's filled in vials or syringes and then put into the distribution channel. So yes, it can take upwards between 7 and 12 months. That vein-to-vein concept, Amy, where a donor could donate at the beginning of January and perhaps we don't have a medicine to the beginning of the following year. Great question.
Well, and Bill, I think that's so important because when you think about pill and tablet drugs or small molecule drugs, that companies can go and source those, the ingredients they need in a lot of different places. And that's even true with cell lines for biologics, but it's so different when you're talking about, you know, you mentioned the hundreds of donations that are needed to just create a therapy for one patient. So
absolutely,
yeah, that concentration is the intense raw material is so important. You talked about healthy human donors and the importance of that, and we're talking about biologic as a starting material, right? So when you think about viruses, obviously, and we talked about COVID, and there are other viruses that were covered by the presentations. Removing viruses is really critical. So can you just talk a little bit about the safety margins involved with viral inactivation for the therapies that our companies create?
Amy, so thank you for the question, and I'll ask others to comment as well. But again, it starts with a healthy donor, right? So, I mean, these are physicals that we do on our donors, and that's the first step in the process. I think importantly, message, there's a robust testing process for these plasma medicines. But so before they're ever introduced into manufacturing, there's testings looking for things like HIV and hepatitis C. And we're making sure that those donors do not have any of that within our raw material before, again, after an inventory hold, it's introduced into manufacturing. I think most importantly for this audience today, what we want them to walk away with, it's been decades where there's been no, and again, that's no documented cases of viral transmission, even though there's a theoretical risk of any plasma-derived protein medicines. So I'll pause there, Amy. I'll ask others to provide color, but that's the brief response to your question.
Thanks, Bill. Debbie or Milan, any thoughts on that?
Yeah, thank you, Bill. I mean, I completely agree. Our plasma protein therapeutics have never been safer. We have thought through this process for many, many years and continue to evolve our processes to ensure that there goes to an infinite zero risk of transmission. But I think what's most important is, the need for healthy donors to come in and donate plasma and to continue to donate plasma and have that commitment. This is a really critically important piece. And, these are the individuals that are committed and we heavily rely on that also help continue to make these products safe.
Absolutely.
And just to comment on Davis and Bill's input, I think that's the commitment from the Alliance members that we stick not only to a high standard, we will evolve it over time, because we can't afford that the therapeutic solution creates an issue. So that's something that's in the process itself, but it's a time-consuming process, but I think it makes perfectly sense to have a stringent focus on this one.
Thank you. Debbie, you talked about obviously a lot of different disease states that are treated, but I think one of the workhorses of the plasma protein therapies is IG. Will you please talk a little bit about kind of the future potential for IG treatments?
So most of the immunoglobulin therapies that are evolving today are really tied to immunomodulation. So individuals with a number of different autoimmune syndromes in which their own antibodies are not working the way they're supposed to and somehow fighting against the individual rather than helping them. So really all autoimmune diseases have some potential in therapeutic potential with the immunoglobulin space. However, there's very good other medicines that are available, and those diseases can be managed very, very well. So there's a continuum of research across autoimmune diseases and severe illness, in which the theory of when antibody therapy can potentially support and mitigate that disease. I would say the large majority of research going on today is mostly in the neurological conditions. And you can understand why that would be such a focus, because the immunologically mediated neurological diseases are so unbelievably debilitating that anything that we could possibly do to mitigate those disorders needs more and more research and more effort. So I would say, though, I mentioned a number of the neurological diseases in which there is approval for immunoglobulin treatment. There are many others that are being added to that list each day. And I think there's very good and strong clinical research that will continue. So over time, we may have more therapeutic opportunities for these individual diseases, which means we need to continually make sure that we have enough immunoglobulin available for those patients.
Yeah, it's a great point. I mean, and especially now that the need has never been greater for a collection. I think what you just said raises an interesting question. There was an article that was recently published in Transfusion that indicated that the clinical need for plasma therapies is continuing to increase. We know that there's been of the pandemic, there has been declines in plasma collection, and the article suggested that there may be a need to prioritize use. I'm interested, we have two doctors on this panel, about your thoughts on that issue.
So I think physicians always consider prioritization of use of rare resources. And of course, immunoglobulin therapy still is considered a rare resource because we have dependency on the donors that come into play. There's no doubt that antibody deficiency has the highest priority for patients because there's really no other alternative therapy for those individuals if they do not make adequate antibodies to protect them from severe infection. However, I do believe that many of these disorders are absolutely necessary, and we really need to rely on the medical community to think about how they use immunoglobulins and are thoughtful about the process, and that they use all of the immunoglobulin opportunities that are out there in their community that are available to them. So I hope that over time we can continue to get the word out to make sure that people understand how important these therapies are, that we can get adequate number of donations so physicians aren't put under the pressure of making a very, very difficult choice and rationing therapy. We know this has happened in the past, but we really encourage the community to help us not get there again. And we look forward to continuing to support those efforts.
Thank you. Thank you. So obviously COVID is on the minds of everyone these days. And Milan, I think you clearly showed the differences between the use of different uses for convalescent plasma. I'm curious though, if you could explain, our PPTA member companies have invested huge sums of money in and commitment and time into clinical trials looking at the development of hyperimmunes. So can you kind of expand on the advantage of hyperimmunes over convalescent plasma or for transfusion?
Thank you, Amy, for the great question. There are two aspects. Let's start with volume discussion. So basically, we have comorbid patients, and every infusion, additional volume is a potential risk, specifically for cardiovascular patient. So if you have the opportunity to reduce volume, it's okay. So I think that's one of the, for me, pertinent benefits. The other one is the level of concentration of acting ingredients. So, and there is a correlation between concentration and effect. Yeah, there the literature is interesting on this one. So if you combine those two, you have for a patient at risk, you have an effective way without increasing the risk for comorbid issues. So it has, of course, a price, and the price is a very clear process in order to achieve this benefit. But I think it's worthwhile, without denying that what Debbie said before. We need people who donate, and we need people who are recovering from COVID to donate their plasma, because as long as we don't have widespread vaccination, and we have some very good experimental drugs, I think that's a valid way to treat a larger part of the population with COVID disease, severe COVID disease.
Yeah, and I think when we think about the development of hyperimmunes, of course, this involves, each therapy involves many more donations, obviously, than a single one-to-one ratio for transfusion. And I'm wondering if you'll talk about the kind of, there's then a broad spectrum of antibodies when there's multiple donations involved. Can you talk about the therapeutic effect of that?
Ideally would be, we have one specific antibody with a neutralizing effect. No, it's not always given that you find it. That's the reason why in those concentrated plasmas, you have a mix of antibodies. But the higher the concentration of specific acting for a specific disease, the better the probability. So that's the reason why those concentration processes leads to a solution with a higher concentration level of neutralizing antibodies for one disease.
All right, thank you. So I think, Bill, I'm going to throw you a softball, but it's a money ball, and this is a really important. I think everything, obviously, this is focused on plasma, and you know, we want the message to be very clear that plasma donation is important. Plasma donors save lives. Why is it important that we collect more plasma today?
Yeah, no, great question, Amy, and thank you. And I'm sure others will have comments as well. And I would suggest after decades of being around the Plasma Protein Therapeutics Association, we all know that there remains a tremendous unmet medical need for plasma-derived proteins across this globe. So I think we need to remain steadfast, as been mentioned by Debbie and Milan today, that we need to raise awareness, educate, and continue to encourage people to donate plasma at increased levels, because there's an existing patient community from a various, and you've heard about all the different disease states today, that are relying on that effort from industry to do that. But in fact, it's been, as you know, just the tip of the iceberg to what this industry can contribute to the health and well-being of patients worldwide with some of these rare and serious diseases. So I'm going to pause there, but it's our passion for the patient that keeps us going. And it's our continuous commitment to those that have been diagnosed and currently relying on these medicines today. And equally important is we know that there's others around the globe that are yet to be diagnosed that are counting on us to come find them and to be able to treat them with these plasma-derived protein therapies. Great question, Amy. We all have a passion for this one.
Yeah, that's absolutely true. And I think when we think about our strongest partners, the voice of the patient is really everything, because it's why we do what we do. And I think no one's message resonates stronger than that of the patient. So when we think about what patient groups can be doing to promote plasma, and we know that certain groups, IDF has a Plasma Heroes campaign, which is has been terrific. But what can patient groups be doing to get the message out that about this? And also, you know, circling back to the question that we talked to Debbie and Milan about earlier, that those decisions about plasma protein therapy should remain in the hands of their medical care providers.
Yeah, I think we'd all say, I'd start with the latter, that we all covet that the value between the healthcare professional and the patient. And we recognize that the healthcare provider and the patient should decide which plasma protein medicine is most appropriate for that particular patient. With regards to the patient groups, it's clear. I think we need to pause here and say thank you. They're tremendous at what they've already contributed. You mentioned the Immune Deficiency Foundation and the Heroes campaign. That's one example of many. All we would say here is if you can do more, or if the PPTA could support you in doing more, our emphasis, like we've said today, is on raising global awareness and providing education on the therapeutic benefit of these medicines, and that we need all hands on deck to deliver. So I'm going to pause there with that question, but it's very clear that the Immune Deficiency Foundation, like others within the Plasma Users Coalition and patient groups worldwide, we just need to continue to encourage them to do more of the same. Excellent.
Great. So I think that this has been a huge learning experience. And I wonder if there's anything further from the group. Any final thoughts?
Yeah, Amy, I'd like to add on to Bill's level of enthusiasm. I think we are at a time in a current pandemic and health crisis to bring these issues forward and to recognize what humans can do for each other and help each other and support the health of many that are less fortunate, that need therapies from plasma protein therapies. And I think this is the time in which, you know, discussions like this are ready to be brought into families, to the dinner table, and to talk about how individuals can step up and be part of this great process to really help those in need. So this is a time of tragedy in our countries and in the world, but there's something that can come out of this that I think will be very, very positive by raising awareness that, you know, this is a very, very precious entity that's needed and will be needed for many, many years to come.
Right, beautifully said. Milan?
I'd like to add on David's comment. The science shows now clear signals that early use of convalescent plasma or immunes have a beneficial effect. Yeah, I'm just citing a recent data set which comes from the US. It's the expanded access program for the treatment of COVID-19. It was just recently published in August. And this is really, really encouraging because not only a lot of patients have been included into the study, we talk about 35,000 patients, yeah, but it shows clearly when you start early to give a plasma therapy at high concentration, you save lives.
Amen.
Yeah, that's a perfect message, and I think it's a great way to end our session. Really, thank you so much, Milan, Debbie, and Bill. This was terrific. You've educated us so richly on this vital issue, and I want to thank our audience members for your engagement. We're at a crossroads, and the world needs plasma. So I encourage everyone watching to visit donatingplasma.org. If you're a healthy, qualified adult, please consider donating plasma. And if donation is not an option for you, use your social media muscle to share this message and get out the word that plasma is needed right now. Everyone is looking for a way to help, and this certainly will help the patients that rely on these products. It's needed to ensure that we can meet patient clinical need. So patients are counting on us, and we're stronger together. Thanks for joining us today.